Today, we are going to discuss the role of vitamin K in blood clotting, and then we will talk about the implications for individuals taking blood thinners such as warfarin. Let's start by discussing hemostasis, the process by which the body keeps our blood stable, or in other words, how we keep from losing too much blood when we get a cut or other injury. When this happens, the body triggers what we call primary hemostasis, in which two things happen. One, vasoconstriction, and two, the formation of a platelet plug. Let's take a closer look. Recall the anatomy of a blood vessel. Starting from the outside, we have the tunica externa, which is mostly collagen. Then we have the tunica media, which is the smooth muscle layer. And finally, the tunica intima, which is made of endothelial cells attached to a layer of subendothelial collagen. Zooming in even further, let's take a look at what happens at the cellular level when an injury occurs. When damaged, these endothelial cells release something called van Willebrand factors, which attract platelets and cause them to adhere to the site of injury, forming a platelet plug. If the injury is small, like a paper cut, reduced blood flow caused by vasoconstriction along with the platelet plug may be enough to effectively stop the bleeding. However, when the injury is more extensive, the platelet plug needs reinforcement, which it gets through the mechanisms of secondary hemostasis. Secondary hemostasis is the process of blood clot formation, which involves multiple proteins known as clotting factors that activate each other in what is known as the clotting or coagulation cascade. The clotting cascade involves two pathways known as the intrinsic and extrinsic pathways that are triggered almost simultaneously and must work in tandem to produce a blood clot. The intrinsic pathway is not the first pathway to be activated, but it is the more complicated of the two and is largely responsible for amplification of the process. It is triggered when exposed collagen in the blood vessel comes into contact with a negatively charged surface, such as glass, and activates the first clotting factor in the cascade. This is known as contact activation, and for this reason, the intrinsic pathway is also known as the contact activation pathway. Clotting factors are generally designated by Roman numerals. The first factor in this pathway is factor 12, which is converted by this contact into its active form 12A. Factor 12A activates clotting factor 11 into 11A, which in turn activates clotting factor 9 into 9A. Factor 9A activates 10 into 10A with the help of 8A. Factor 10A converts prothrombin into thrombin with the help of factor 5A. Thrombin then activates fibrinogen into fibrin, which forms a mesh at the injury site. Thrombin also functions to amplify the effects of the cascade by increasing production of factors 5, 8, and 11 in order to produce enough fibrin quickly. Additionally, it activates clotting factor 13 to 13A, which forms cross-linkages that help stabilize the fibrin mesh. The extrinsic pathway actually initiates the cascade and is triggered when damaged tissue releases something called tissue factor. Tissue factor combines with factor 7A to activate factor 10 into 10A. This then continues along the pathway to form thrombin, and so on. Now, you may have noticed that factors 5 and 8 don't seem to have been activated by anything. Interestingly, these factors don't function the way that other clotting factors do. Instead, they function as what are called pro-cofactors. Rather than circulating in the blood like the other clotting factors, factors 5 and 8 are found on the phospholipid surface of the activated platelets that moved to the injury site during primary hemostasis. This means that clotting factors 9 and 10 will not be activated until they are at the site of injury, where factors 5A and 8A are located, ensuring that a clot forms only where it is needed. So where does vitamin K come into all of this? Vitamin K also functions as a cofactor in the clotting cascade, helping to activate clotting factors 11, 10, and prothrombin by serving as an electron donor. It does this by allowing for the carboxylation of specific glutamate residues on the clotting factors. Now, if you remember your OChem, you know that a carboxylation reaction occurs when a carboxylic acid group is added to a molecule with the help of a catalyst. In this case, vitamin K is oxidized, meaning it donates electrons to carbon dioxide and oxygen to form water and a free hydrogen ion. The carbon dioxide in turn becomes a strong nucleophile, which carboxylates the glutamate residue, forming gamma-carboxyglutamate. This is key because gamma-carboxyglutamate is able to bind with calcium, which then neutralizes its net charge. This neutral charge is what allows for the clotting factors, particularly thrombin, to bind to each other, resulting in a clot. Now, what does this have to do with warfarin? Well, warfarin is an anticoagulant, which is prescribed to help prevent blood clots in those at risk of heart attack or stroke. It works by interfering with vitamin K recycling. This is the process by which the vitamin K epoxide that was formed during the carboxylation reaction is processed back into vitamin K through a series of 3 different enzymatic reactions in order to be used to continue to support the clotting cascade. If less vitamin K is available, then clotting or coagulation will be reduced. The important thing to remember is that warfarin is dosed based on vitamin K levels to produce a balance between clotting and no clotting. Too much vitamin K and warfarin will not be as effective at reducing clotting risk. Too little vitamin K, and there is a risk that warfarin could lead to excess bleeding. So if you are on warfarin, vitamin K levels must remain consistent to benefit from the effects and avoid these potentially serious complications. Thanks for watching.